Welcome to today's biology class, and we're going to be focusing on the AS topic, biomolecules, and to be more specific, carbohydrates, just carbohydrates. So where do we begin this topic? We have to start with some terms such as polymers, monomers, monosaccharides, disaccharides, polysaccharides, and macromolecules. So first off, macromolecules, it just means a large molecule. That's all it is. Then we move on to the word polymer, which means many molecules joined together of repeating subunits. So basically it means we are taking one particular subunit, one particular item, and continuously repeating it probably in a long chain. While monomers are those subunits that we're talking about in a polymer. Now let's talk about monosaccharides, disaccharides, and polysaccharides. These are all levels of sugars, because saccharide means sugar, while mono, di, and poly refer to the number. So monosaccharides are simple sugars. These are basically just your one subunit. These are the basic subunits, and you probably know the regulars like glucose, for example, C6H12O6, and we tend to characterize them by the number of carbon atoms that we find. So for example, a sugar with 3 carbon atoms will be referred to as a triose, a 5 carbon sugar as a pentose, and a 6 carbon sugar like glucose as a hexose. And if you remember, pentose such as ribose and deoxyribose are commonly found in nucleic acids such as DNA and RNA, but that comes in for another topic. Now let's talk about what we notice with glucose in particular. Now we're all used to C6H12O6, but when you enter your first year of A levels, you tend to notice that you learn that glucose can appear in a ring form, and it does so in 2 isomers, if I can say that, and that is alpha glucose and beta glucose. Now the main difference which you need to understand between alpha and beta glucose is with reference to carbon atom number 1. In alpha glucose, you see the OH group attached to carbon atom 1 to be at the bottom in reference, and when you get to beta glucose, we see that carbon within that same carbon atom number 1, we see the OH group on top. Now this might not seem like a huge difference, but it plays a very, very vital role when we talk about forming polysaccharides, especially when we're talking about maybe starch and glycogen against, let's say, cellulose, but we'll get to that at the end. Now let's talk about disaccharides. Disaccharides, as the word shows, di, which means 2. We're talking about 2 monosaccharides or 2 subunits being joined together. And a very important thing for you to notice is the name of the bond being formed and how it's formed. So 2 glucose molecules can be joined together, for example, to form maltose. And the bond formed between these 2 molecules, or between 2 monosaccharides, is known as a glycosylic bond. This glycosylic bond is formed by a condensation reaction, which means that water is being removed, and this is done by using the OH groups that we find in our sugar molecules. Basically 2 OH groups can enable us to remove 2 hydrogens and 1 oxygen molecule to form the water that we're going to remove. Now this also forms with things like lactose, which is glucose and galactose, and sucrose, which is fructose and glucose. So those are the basic disaccharides that you probably should remember, but by the time you probably get used to them, because as you remember, sucrose is the same carrier sugar or carrier transport sugar that we notice in plants. And basically that's it for disaccharides. Talking about reducing sugars and non-reducing sugar tests, that's going to be the next video. So I feel like it's better we talk about that separately, because it's a very, very interesting part of the topic that comes out not only in your objectives but also in your practicals. Now let's talk about polysaccharides. When talking about polysaccharides, we're going to focus on 3 major polysaccharides for the sake of your A levels, or AS in particular. We're talking about starch, glycogen, and cellulose. Now with starch in particular, starch is made up of 2 substances. one known as amylose and the other which is amylopectin. It's very key that you remember these two because they tend to be different in terms of structure. Now, let's start with what's common before we get to what's different. Amylose and amylopectin are both made up of alpha glucose molecules. That is something for you to note. Alpha glucose molecules. So starch is made up of alpha glucose molecules. With amylose, we tend to notice that we have an unbranched chain that tends to form a spiral helical shape at the end of the day, and is formed by having a glycosidic bond that is characterized as alpha 1,4 glycosidic bonds, which means that the carbon atom number 1 and the carbon number 4 from another molecule are providing the OH groups that we're going to use to draw out water in this condensation reaction. So alpha 1,4 glycosidic bonds, that's what you tend to notice in amylose, because it's telling you what number carbon atoms your OH groups were used for the condensation reaction. So you tend to have an unbranched, that's a very key word, an unbranched chain of alpha 1,4 glycosidic bonds within the molecule with amylose. And when you get to amylopectin, we have a branch structure. So you have the regular alpha 1,4 glycosidic bonds, but we also tend to have alpha 1,6 glycosidic bonds. So imagine you have a sort of, not straight, but an unbranched chain, such as amylose. Then you notice a bit of few projections along the way, maybe up or down, whichever way. That lets you know that it's a branch chain, and for amylopectin, it tends to be alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. And these two together form, amylose and amylopectin form starch, which you notice is a storage form of sugars in plants. Now, when we go to talk about glycogen, which is a storage sugar in animals, or mammals such as us, we notice that we have a branch chain very similar to that of amylopectin, meaning that we have the same alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. So you need to remember, glycogen 2 only utilizes alpha glucose molecules. And it's very key because now, what's the major difference between amylopectin and glycogen, since we said they are quite similar? Well, is that we notice that in glycogen we have far more branching. The branching in glycogen is much greater than that of amylopectin. So that's just the major difference in that case. And once you understand starch, it's very easy for you to characterize glycogen when explaining it. Now let's talk about cellulose. Cellulose is where things get interesting when it comes to polysaccharides. With cellulose, we notice that it uses beta glucose molecules. In cellulose, we have an unbranched chain, but you notice that it doesn't form the exact same way as you would see with, let's say, amylose in starch. It doesn't give you that straight alpha 1,4 sort of movement, but here we have beta 1,4 glycosidic bonds. And because of the certain way that we said previously, that the OH group on the carbon number 1 atom is sort of flipped. It goes from being at the bottom, from being under to the top when drawing it. You notice that you have to flip every other molecule, beta glucose molecule, for you to have that same 1,4 chain. So that's one major difference you'll notice. When it comes to cellulose, you tend to have every other beta glucose molecule flipped by 180 degrees to enable us to have that nice looking unbranched chain. Now let's talk about cellulose in terms of its function. Now we know cellulose to be in plant cell walls, and obviously we know it provides rigidity and strength to the cell wall. Now, how does this happen? If you line up two cellulose strands together, two unbranched chains, they are held together by weak hydrogen bonds due to the presence of the OH attachments to the carbon. Now, we've said this is weak, but we talk about it being strong and providing tensile strength to cell walls and to other organisms and to other parts of whatever it's involved in, you see that by the time you compile about, let's say about 60 to 70 of these chains with all weak hydrogen bonds, it tends to become stronger. And these are called microfibrils. But when you take these microfibrils and you take more and more bundles held together by these same cross links, you tend to see that you have a very, very strong bundle. And these fibers are what provide the high tensile strength noticed with cellulose in different things such as the cell wall of plants. So I hope you found this video really interesting. I hope it's helped you a lot. If you found it helpful, please consider subscribing, like, leave a comment. Let me know what other topics you want me to cover next in case you need that for school. Yeah, thank you for coming to class. Yeah, that's kind of weird.